Welcome to the Trailblazers. I'm your host, Perry Rarick. The purpose of the Trailblazers is to share with you interesting discussions I have with trailblazing business leaders. My guests are innovative, they're experts at what they do, and they're not afraid to challenge the status quo when there's a lot at stake. My trailblazer guest today is Dr. Alexis Santana, a molecular geneticist and cancer immunology researcher with extensive experience in organoid technologies and translational oncology platforms. She earned her PhD from Stony Brook University and completed postdoctoral work at NYU. She also completed an executive MBA at Rutgers Business School. Interesting combination there. After her academic research career, Dr. Santana transitioned into scientific sales and business development across several contract research organizations, or CROs. She worked with pharmaceutical and biotech teams to support the adoption of organoid platforms, preclinical oncology models, and advanced biological profiling technologies. She currently works at Samsung Biologics, where she supports commercialization efforts for the company's newly launched patient-derived organoid platforms across the US and Europe. Alexis, welcome to the Trailblazers.
Thank you, Perry. I really appreciate the invite, and I'm excited to talk about organoids today.
And I am as well. Very interesting topic. And until I met you, I didn't know what an organoid was, but it's very, very interesting. So, back in December of 2022, the US Congress passed the FDA Modernization Act 2.0. And from what I can tell, it eliminated the long-standing mandate for animal testing in new drug applications. That was followed by the FDA outlining their new approach methodologies in April of 2025, not very long ago, identifying a number of innovative scientific tools and approaches to reducing animal testing in preclinical safety studies. Patient-derived organoids, uh, of which you are an expert in, is one of these tools, and it's the topic of our discussion today. So, this is a fairly new service that's being rolled out by Samsung Biologics. And if you could help us understand this solution in the large, very complex process of bringing a drug from discovery all the way to commercialization, where do these services lie?
Yeah, great question. So, Samsung Biologics was founded in 2011, and we've served traditionally as a contract development and manufacturing organization. And so, what that really means is that we have focused on later stages of a drug's life cycle, mainly to handle its development all the way to the large scale manufacturing of the drug. And so now we've moved into the research discovery preclinical space by launching Samsung Organoids. And so that really expands our end to end capabilities across the entire drug life cycle. So we're, covering early all the way to late.
And so how common are organoids? Or have I been living under a rock and I just recently heard about these, but how common are organoids?
Yeah, organoids, they've traditionally been common in academic settings where folks were utilizing them prior. I would say that there has been an uptick in the use of organoids, predominantly at the pharma R&D stages, where a lot of groups are utilizing them for efficacy and safety testing. Um, this is really largely driven by the NAMS announcement that you just described, where the FDA is actively recognizing and encouraging the use of organoids, microphysiological systems, and computational models. And a lot of the rationale for this push is due to them being better at reflecting human biology, but also just to reduce reliance on animal testing.
And so you mentioned that they've been around the academic communities for a while, less common in CROs and CDMOs. How many CDMOs, CROs have this capability?
Yeah, so it's a very highly specialized process for the establishment and testing of these organoids. Traditionally, these studies are run out of CROs. You will find a couple, I would say a handful, to my knowledge, of forward looking CDMOs that are offering this service, like Samsung Biologics.
Okay. All right. So, how do they work? Can you describe how are they used? How do organoids work?
So I see this, I really like this question. I see this as a three-step process. The first step is to establish the organoid model for the disease that you're looking to test, if it's disease or non-disease. They can be established directly from patient biopsies or from induced pluripotent stem cells or iPSCs. And so these are cells that are obtained from patient blood or skin samples that are cultured into a stem cell like state. And then they're coaxed into developing into an organoid through additional culture methods. And so these are really patient specific types of models that you can establish. These organoid models, as I mentioned, they're cultured in very sophisticated manners. So this culture condition is critical to their growth. They're typically grown in a 3D extracellular matrix with a enriched media, and so that's really what you'll do. You'll establish your model, and then you go on to the testing process. And that establishment takes some time, takes a couple months, depending on the type of organoid that you're building to grow, establish, and characterize them for use. The second step would be to run a study utilizing those organoids. And so you can run, I would say there are two types of major studies that are run. So efficacy or toxicity or safety testing using those organoids. Those are typically run in a monoculture assay. You can run co-culture assays. And when I mention co-culture, that's incubating an organoid in the presence of fibroblasts or immune cells that sometimes can be derived from the same patient that you're establishing the organoid from. These monoculture assays are run in high throughput plates. And so these are 96 or 384 well plates. And then what you'll do is you can expose multiple organoid types, so multiple patient organoids or multiple drugs to these organoids in a 5 to 7 day type of assay. And so what you do is you'll dilute the drug and then you will incubate it with the organoids and then measure responses. And so that's the second step, right? So it's culture, culture with drug, and then measure responses. Finally, what you'll do is gets your readout, right? Are you measuring efficacy? Are you measuring toxicity? Are you measuring cell death, basically? So what we'll do is there are different types of readouts that are available. One for an organoid monoculture, folks will typically use a standard viability assay, so cell titer glow readout to just measure, does my drug kill the cancer cell? Does my drug, cause toxicity in a liver organoid or a cardiac organoid that I'm testing? And then we do have some additional readouts available. So you can use high content imaging to look at structural changes in the organoid upon drug treatment. You can track drug internalization. So for folks working on ADCs, you can look at how your drug internalizes into the organoid. And for co-cultures, which I mentioned early on, this is kind of like, as an immunologist, this is my favorite part of the process because you're treating an organoid with a drug, and you can use co-culture assays to traffic, let's say, immune cells into that specific organoid. And so we can use high content imaging to look at that trafficking. You can measure cytokine release. So are the immune cells active in secreting cell killing cytokines? And then you can also profile the cell types within that organoid. So within that organoid co-culture. And so there are a bunch of different types of assays that you can use at the endpoint. I will say that one thing to really consider here is that that first step, so establishing the model, takes a long time. And that's one of the advantages of partnering with companies such as Samsung Organoids, is that instead of spending months and months on end to establish and characterize your model, we have already established a biobank, we've characterized it, and you can go from straight into testing with your drug to final data within about a 5 to 6 week period. So For using outsourcing this type of service, you can get data within about a month.
You speak about this with such ease, and to me, it's amazing. It is really amazing, incredible stuff. In a moment, we'll get to that first step, that challenging setting up the model. I'm gonna have you talk a little bit more about that. But I want to, at a high level, at a broader level, one of the benefits of this is it eliminates animal testing. What are some of the other big benefits, or sometimes I like to refer to them as chronic problems that the pharmaceutical industry faces? What does this help overcome?
Great question. So, organoids are positioned to help tackle the translational gap. This is often encountered by the pharma industry, where about 90% of drugs that look good in cell lines and animal models will ultimately fail when they reach the clinic. And so, what happens is this results in decades of work and billions of dollars that are lost because these models did not reflect human biology early on. So organoids really help to avoid these failures by reducing that attrition rate and providing a human filter before these drugs get to the clinic. So really, instead of running lengthy animal studies or having those failures at late stages, you can run an organoid study in about a month and get data that helps to inform and de-risk your pipeline. So some examples of this are the use of liver organoids, which the FDA has recognized. These are good for, safety and toxicity testing. And liver tox predictions are at about 90% accuracy. And so they really can help the pharma industry flag toxic drugs that would only typically appear in those costly late phase 2 and phase 3 clinical trials, and so that's one of the utilizations of organoids. On the oncology side, at Samsung Organoids, we are growing organoids from patient tumors, and so because these organoids are derived from individual patient tumors, this permits us to kind of get into the personalized medicine realm where you can test multiple drugs across one patient to identify which drug would work. Or what you can do is screen a large panel, let's say 25 to 40, 50 patients, to see where your drug would fit if you're looking to expand indications or repurpose a drug that you're working on. And then, lastly, data. It's all about data today. So integrating the sequence data, clinical data that's obtained on these organoids and using bioinformatics, it's really possible to start to drill down on patient populations for targeting for clinical trials. And so organoids bridge a gap, and that's that translational gap that enables faster, more accurate, and personalized drug development for pharma.
Yeah, you're really connecting the dots for me now. It's really, it's very promising. I can see that now. And your talk about, it's gonna save time. Time is money. People are sick. There are patients at the end of this, and it really sounds promising. I wanna go back to something you said earlier, about that first step, modeling, being so challenging. Why is that?
Good question. The modeling is challenging. I could see a couple different ways in which that challenge could exist with the organoid system. First is that they lack a native immune system and blood supply. So most observations that you're making are within the context of the organoid itself. I did mention that you could utilize co-culture systems with PBMCs or other cell types to kind of get around that. And I've also seen some very interesting data on organ on a chips where you overlay endothelial cells and grow vasculature in these chips. And then you can show some trafficking of immune cells into the organoid system. So we're getting better at starting to model the microenvironment of the organoid or organ where these models are coming from, but we're not 100% there yet. Reproducibility is also a significant challenge. So I mentioned really early on that culture conditions are super important. And since they're so sensitive, if there's some variability in your culture conditions, that could lead to batch variations. One way that we mitigate this at Samsung Organoids is through the maintenance of a rigorous QA QC process. We work within standardized workflows, and you can't, you know, pass these organoids out into infinity. We try to maintain them at low passage just to, retain the molecular fidelity of those organoids. And, there are a couple, maybe two more things that I think are really important. And I mentioned this early on. You're obtaining clinical data, you're sequencing these organoids, you're characterizing them very early on. You have so much data that you need to analyze, and then you're treating them with drugs. So you have to integrate all this in a system, and this requires really sophisticated bioinformatics. And so you will need some level of bioinformatics support or bioinformatician on staff to be able to really make meaningful insights of the data you're generating on these organoids. And then lastly, growing a 3D model is not the same as growing a 2D cell line on plastic. So you will need a very sophisticated lab. When I talked about culture conditions and batch to batch variability, this some of this can be overcome using automated liquid handlers that can read and analyze multi-well plates in high throughput. And so those are the challenges that I think need to be considered when it comes to organoids.
Amazing. So I'm a drug innovator. What stage of development am I in that would make your services, make me and my molecule a good candidate for your services?
So I think early stage discovery preclinical drug developers would benefit from the organoid service, predominantly because you're thinking about the efficacy of your drug and the safety of your drug. So those will be the types of uh, folks that would be interested in running organoid studies.
All right. So this is, we mentioned at the beginning, Congress passed the FDA Modernization Act. The FDA then produced some announcements about new approach methodologies. Organoids are one of them. How does the FDA feel about this? How are they working with you and other companies to advance these approaches?
That's a good question. The FDA, as I mentioned, is actively encouraging the use of organoids as a NAM, but they are definitely evidence driven in that encouragement. And so, since they announced the FDA Modernization Act 2.0 and their 2025 roadmap, they made it clear that they want animal testing to remain the exception rather than the rule. And organoids sit within that NAM space and are being viewed as human relevant tools for efficacy, safety, and toxicity prediction. We actually saw this crystallize earlier this year, very exciting. In 2025, the FDA approved an oncology IND for a drug utilizing solely vascularized tumor organoid data. And there were no animal models that were utilized or animal testing reported to be utilized within this IND application. So they're willing to accept organoids for efficacy testing right now. And I think safety and tox is gradually moving in that direction. But they really see this organoid system as a complement to animal safety data.
Excellent. I want to switch. You're responsible for the commercialization of these services for Samsung Biologics. And I want to talk a little bit about that. This is a little different. This isn't, we've already covered, this isn't really common. Although the FDA, everybody seems to be excited about this, reducing animal testing. And also just some of the other benefits that you highlighted of this testing, timelines and whatnot. How do you communicate this? And this is very different than Samsung's other capabilities, which are tend to be on the commercial manufacturing of biologics. So, how do you go about communicating this to an audience that may be unaware of what organoids are and the benefits?
Yeah, I mentioned this early on that use of organoids has predominantly been in an academic setting, and it's starting to pick up. It's an emerging need for clients. So the biggest challenge, I think, as you mentioned right now, is just that a lot of teams are still new to this technology, and they're naturally going to ask, "Can I trust this data? I'm making high stakes decisions. People's lives are at hand. Can I trust this?" And so we bridge this by taking a consultative approach using education and evidence to kind of bridge that confidence gap. And what we do is we lead with data. We share white papers, posters, publications from peer-reviewed journals, and we also do a fair bit of R&D in our own labs to help and use that to build case studies that help to show the translational relevance of our models. And then again, we're a very flexible organization. So every client interaction is more about engaging with our scientists to help explain any nuances or questions that a client would have. And then also helping to interpret the science later on after running an assay. So I think we take a consultative approach, we use education, we use evidence, and that shifts us from just being a vendor to being more of a strategic partner that helps our clients to de-risk their programs.
Yeah. Excellent. That's a different type of, I'll use the term marketing. The way you make an external audience aware of your capabilities. That's not about size of bioreactors and capabilities and how quickly you can take on work. This is a deep, deep conversation, a very give and take discussion with your potential clients, I assume.
Yes. It's about trust building.
Oh, I love to hear you say that. It's true. Yeah, building trust, which is in this industry is so important between client and service provider. And I don't even want to say service provider, partner, strategic partner. It's interesting. So when I was preparing for this conversation we're having, most of what I saw was about safety testing, elimination of animals, all about, but you have brought up efficacy a number of times. This goes beyond simply a safety test. So there's more to this, I think, in the future. Can you talk about where this is going and how far can this go in actually predicting efficacy of some of these drugs?
Thank you, Perry. This is a great question, and I am so excited about the future of organoids. And so, in my own personal opinion, first, I really believe that organoids and other microphysiological systems or organ on a chips will become a standard part of preclinical packages, particularly in areas like oncology and liver, maybe cardiac safety testing. I will say that FDA is certainly playing a role in driving this adoption at these early stages, and so there's support on the regulatory side for this. I do want to mention though, that this will not completely eliminate animal testing. And so I do anticipate that there will be some models that will eventually be retired. And that's really particularly in cases where these organoid systems themselves provide, more human relevant data. And then what I'm really excited for is something that you can only dream about as a graduate student at Stony Brook, or even something we were working towards when I was at in my postdoc at NYU. And it's really the idea of building patient avatars. And so because you can grow organoids from an individual patient's biopsies, or even grow them from blood or skin cells, so derived from iPSCs, as I mentioned earlier, we can start to establish these biobanks of patient samples. And then by giving clinicians access to this, they could potentially screen drugs in these avatars prior to moving into a patient. And so that would really increase the chances of efficacy for that patient and safety for them. Throughout that process, it even can be utilized, and we've seen some of this happening in the context of clinical trials. And so, helping to match patients with clinical trials utilizing some of these patient avatars and testing on those. And then what's really exciting is because are in this AI and digital age, I can see a lot of the digital technologies that are being utilized in many different types of companies, including, omics, spatial transcriptomics, high content imaging, CRISPR. and AI computational models layered on top of these organoids, with this data to be utilized to build digital twins of organs or even individual patients. And so that's personalized medicine. I've seen a lot of companies coming out utilizing AI for drug discovery, utilizing AI or spatial transcriptomics to develop diagnostic tests or even match patients with clinical trials. And so I think there's, utilizing all this data, I think the future is really comprised of an ecosystem with organoids at the center of that. Basically, what people are doing are using data to develop hypotheses, helping to whittle down on the drug discovery cycle, helping at those late stage clinical trials. And so that these organoids at the center of this, helping to provide a more human centric way of developing and personalizing medicines. And then hopefully, all of this combined will help to reduce the attrition rate and that translational gap that I described early on.
Amazing. Patient avatars. I mean, this sounds like science fiction to me, but it's happening right now in such a promising future. And you're leading the way. So thank you for being involved in this. And it seems like you're at the right place at the right time with all of your background. This is very exciting stuff, and I hope we can catch up again down the road to hear about updates and all the advances you're making. Alexis, how can our listeners get a hold of you?
Yeah. So thank you, Perry, for having me. I hope this was helpful. I'm happy to connect with folks to follow. Feel free to connect with me on LinkedIn. We can start a conversation there, or you can also submit an interest form on the Samsung Biologics website.
Excellent. Well, my guest today has been Dr. Alexis Santana. She is a molecular geneticist and cancer immunology research. She's an expert in the employment of organoids, such a fascinating innovation that's taking place right now. She is a trailblazer. Alexis, thank you for being part of the Trailblazers.
Thank you for having me, Perry. It was great to be here today.